So we just discussed the importance of the structure, or at least the primary structure of DNA. And we said that along the backbone, this backbone will contain sugars, and I'm going to abbreviate that with an S, that's connected to a phosphate. And then another sugar that's connected to a phosphate. And then another sugar connected to a phosphate. It goes on and on and on and on. And then off of this sugar backbone, this is where our nitrogenous bases will come into play. So here is where the A, T, G, or C will come into the picture. Keep in mind that if this is RNA and not DNA, the T gets substituted for the U. As well as the ribose, if we look at the ribose or the sugar part, all of these typically will have a substituent on them or a group that is hanging off. And one of those groups that's pretty important is right here at the 2' position. And that 2' position, there will be an OH present if this is RNA. And if this is DNA, the RH will not, or the OH will not be there. Okay? Another thing that we talked about as well is that the phosphate end will be given the 5' end or location, and the OH group will be given the 3' end. We also said that whenever we write the primary structure of the DNA molecule, we start with the 5' on the left and the 3' on the right. We also do not write the sugars or the phosphates that are there. We simply write the bases that are present, and that is it. So you'll just see a series of letters most of the time when we denote the primary structure of DNA. All right. So now what I want to do is I want to go and talk about the 3D structure of DNA. We're only looking at primary here, and that is it. So we want to make sure that we understand fully this concept of how DNA is going to order itself and twist around and come into this 3D version that we always associate with the structure. And there were a handful of people that were in charge of this determination, but there's 2 people that really got the credit for it. And this was James Watson, you can see his name here in the middle of the screen, and Francis Crick. And they were the ones that came and published a paper and were really the first ones that said, here's the 3D structure for a nucleic acid, or what we will call deoxyribonucleic acid, aka DNA. It's still one of the scientists' most important discoveries. That's typically how we still refer to it up to this point, but it's also one of the major soap operas that happened during the day as well. So James Watson, Francis Crick, they came up with this idea of the 3D structure for nucleic acid. And this happens in about the mid-1950s. So DNA is still fairly new to us, but we have came a long way since the mid-1950s on how to determine it, how to measure it, how to analyze for it, how to compare it. There's so many things that have been done in this field of DNA that it's quite remarkable just what we have been able to accomplish within 50 years since the time that we just really figured out this structure, and that was about it. Okay? So basically what happened is that James Watson, here he is, current age 91. I mean, to this date that this video has been recorded, he has not passed away yet, so he's still with us. Again, this is fairly recent stuff, okay, and this kind of proves that point. But he really kind of helped determine the 3D structure by using cardboard and X-Acto knives. He basically took pieces of cardboard. He knew that the bases were pretty much flat, and he knew that cardboard was flat as well. So why not take these pieces of cardboard, why not cut out the shapes, and then maybe use that as a starting platform from then on out. He was not really a chemist by trade. He was a biologist. He changed his major from the study of birds, ornithology, to genetics because he took more of an interest in it. So if he did not change his major, just think about what could have happened here. But here he is. James Watson had what I'm going to call a partner in crime. And this partner in crime, his name was Francis Crick. And here's Francis Crick from 1916 to 2004. So he passed away not too long ago. Age of death was 88. And he brought something new to the table. He was another biologist by trade, And he really looked at this process and said, you know what, DNA is kind of the core, and this is what we're going to call the central dogma. And if it wasn't for DNA, we cannot make RNA. And if it wasn't for RNA, then we cannot make proteins. That's kind of their job. And again, up until this point, think about it. Most people thought that protein was the genetic carrier, that DNA was not. And now all of a sudden, we get this discussion that DNA is going to be the boss of everybody, that needs to be made, that needs to be sent out into the body, that has a certain role or a certain job to accomplish. It's all DNA leading the charge. His PhD, well, this is kind of a fancy degree that people typically struggle to get, right? And this PhD came from measuring the viscosity of water at high temperatures. Nothing more complicated than that, people. Of course, back in the day, he was generating data that we did not have at the point, so it was fair game. I mean, this was his PhD dissertation. The research happened during World War II. A bomb actually fell through the roof of his lab, and that destroyed his apparatus that he was using at the time. He also really focused a lot of his time on the brain. He wanted to know how the brain operated and what made the brain churn and what made it think and what made it remember and how could we go back and remember memories and that kind of stuff. So how does it make a conscious mind? This was something that he, of course, dabbled into but didn't make too much leeway in. He also became a master of X-ray crystallography, and X-ray crystallography is an instrument that we can use in today's laboratory environment. But way back in the day of 1950s, X-ray crystallography was kind of on the fringe. Not a lot of people knew about X-ray crystallography or how to read it or how to analyze samples with it, but nowadays the footprint's pretty small. We can bring it into a lab. A lot of people can be trained very easily on how to use it. Computer software does a lot of the work, so it makes our job much easier as laboratory analysts. So this X-ray crystallographer machine, or X-ray crystallograph, is going to come around and it's going to help confirm the structure of DNA. So these two people kind of work together. You always hear about Watson and Crick, and you always hear about the discovery that they did. So this is what they proposed. All right, this is kind of a summary of everything that they went through and detailed and provided us documentation on. So number one, up here at the top, is that DNA is composed of two strands. All right, so up here, here's strand 1 and here's strand 2. And these two strands wind together in a right-hand twist. So basically what we're saying is that if this is a ladder and the bases are kind of here in the center, that this DNA will wrap around itself and it will twist all the way from beginning to end. And it's a right-hand twist. The diameter is 20 angstroms, so we're talking about the width, of course, of the DNA strand. The two strands run anti-parallel. So what that means is that if this is 5' and this is 3' on this chain to the left, then over on the right-hand side, it's going to run backward. So this is now the 3' and this is now the 5'. That is what we call anti-parallel to each other. The two strands wrap around each other. Well, we kind of know that. If we're talking about the twist, we know that these things are going to wrap around on each other as well. The bases occupy the core of the helix. So the helix is the shape of what we describe as it twists. And all of the base pairs that are on the inside of those strands, all of these base pairs right here, they are nestled in the middle of the helix. So as it twists like a slinky, think of that at least, there's going to be base pairs that will be here in the middle. And that's how this thing is going to be structured. The sugar phosphate creates the backbone. This is something that we've already talked about at this point. That's not surprising. Of course, it's loaded with OH groups. Think about those sugars. Those sugars have a lot of OH groups that are hanging off. Phosphates also have oxygens on those as well. So this negative backbone, there's electrons on the oxygens of the OH groups. There's also electrons on the oxygens of the phosphate groups that creates a highly negative charge, and this negative charge repels what we call nucleophiles. Nucleophiles in organic chemistry, this is really how orgo reactions begin to take place. We often think about what is the nucleophile and where is the nucleophile going to attach. Nucleophile is basically something that doesn't want to be a charge anymore, and it's going to go out and it's going to find things that it can react with, and they're typically negative. All right? So these negative pieces make reactions. It makes compounds convert from one form to another. Well, the good thing with DNA is that these are loaded with negatives on the outside of the helix. Who cares about the middle? But the outside layer of the DNA is negatively charged all the way around due to the OHs of the sugar and due to the O's of the phosphate. And that is why a nucleophile, when it comes into the presence of DNA, whoop, away it goes, because this thing is loaded with negative, and this thing's already negative, and two negatives repel each other. They don't want to hang out with each other. The bases in the center, they are flat, and they help make this ladder notation that we keep drawing over and over and over. We also have talked about the presence of the hydrogen bonds. So now we know that those are present. Those lead to stability of the bases. Those keep the two strands of DNA together that run anti-parallel. It keeps them kind of nestled together so they can curl on themselves and make this helix. Each turn, if you can see up here at the very top, I've kind of done this squiggle. So as the DNA twists and turns, there's actually on average 10 base pairs per turn, and they align perfectly with each other. Okay, so what does this mean? They align perfectly with each other. Well, as this DNA molecule twists, this molecule is very symmetrical, meaning it's very even top to bottom. So there's got to be a reason for that. And that's because when these two strands come together to make this ladder, these pieces on the inside, the way that this is structured, because they are very specific, we've said A always pairs with T, G always pairs with C. The length of the base pairs that's here in the middle, they are almost identical. Okay? They align perfectly with each other. So when the A and the T comes around and the G and the C come around, then that length is pretty much the same because one is purine and the other one is pyrimidine. If both were purines or if both were pyrimidines, what we would see is that the A and T might be squeezed a little closer together and the G and C might be a little further apart, and then the A and T would come closer together, then the G and C would come further apart, and we would get this kind of curvy structure in DNA on the outside backbone. And this ladder becomes weakened because of that. I don't want to claw up on that ladder that has those runs at different widths and lengths. Do you? No, I don't. So DNA doesn't either. So that's why that this pairing is very important. A and T and G and C go together. One is purine, one is pyrimidine every single time, and it makes consistency all the way through the DNA strand. The DNA strand has a pitch, which basically means how far does it go up as it turns, and that pitch is 34 angstroms. So it's a little bit taller than it is wider, if you want to think of it that way. The diameter of DNA is 20 angstrom, but the rise as it goes up is 34 angstrom every time it makes that turn or that loop. Base pairs can be exchanged without altering the DNA structure. So basically what happens is that if we have an A and a T that's sandwiched inside of a DNA chain, then what we could do is that DNA strand could make the decision to get rid of A and T and then replace that with maybe GC. And this does not alter the DNA structure. The only thing that it has done is it's altered the base pair orders that's on the inside of this molecule as it turns and twists. So that's it. So there's no major disturbances with the stability of the molecule if we do that. And this is really what's going to give rise to different DNA from different people. It also is going to give rise to maybe genetic mutations and stuff. So we can pull out some of these base pairs, replace those base pairs with something else, and now you have a different DNA strand that will give different orders to your body on what to do. So we can replace these without a problem. It's not going to disturb the stability of the DNA chain. DNA also forms what we call minor groove and major groove, and there's a link to a YouTube video here that will kind of depict a lot of these measurements and a lot of this 3D formation that we're going to talk about here in the next few minutes. So that link will be in the Blackboard. Please click the link, watch that external video, and they will go through and kind of describe something with computer models and that kind of thing about this 3D structure. All of these things together are what we're going to call bDNA. And bDNA is going to be the most major form of DNA. As far as the total length goes of DNA, we're talking about how wide and the rise per turn, but DNA can be up to 6 feet long. So just think about that for a minute. I can take a DNA strand from you, and if I have a full strand of DNA and I stretch it out without breaking it, I can stretch that strand out to 6 feet, and your body has tons of DNA inside of it. So that means that this 6-foot molecule, has to be tightly wound up, as tight as it can get it, inside of your cells that carry it. So just imagine how tiny a cell is. So we're going to take this 6-foot-long molecule, we're going to wrap it up, we're going to bunch it together, we're going to cram it into your cells that will probably have more than just DNA on the inside of it. But something has to be there in order for this to happen. And this is something that we call a histone. A histone will allow the wrapping of the DNA molecule, tightly wound it, and cram it into your cells that you carry with you every single day. So when we extract DNA from someone, one of the things that we have to do is that we have to break the DNA free, and we have to release it. And one of the things that we have to do is deal with these histones, because the histones are going to wrap this DNA very, very tightly and cram it inside of your body. So we're going to have to destroy that structure. We're going to have to destroy it so we can free up the DNA that we want to analyze. So this is one of the things that basically has to be sacrificed throughout this process. Again, I've got another YouTube video that I'd like for you to watch. This YouTube video will be on Blackboard. Just click the link, and it will probably relate the histone activity and how it does its job and how it packs all of this stuff up inside of your cells. Other things about DNA. DNA does not have an OH group on the ribose ring, and RNA does. And I keep saying that this is basically due to stability. DNA needs to be more stable. So on this ribose ring, as we start building DNA, DNA has made the decision over a course of time to get rid of that OH and not have it present. The reason is that this OH, again, if you are in organic chemistry right now, is a nucleophile. This has a negative charge that's associated with it. And if it does, then a reaction can happen here. This is a traditional alcohol, and that alcohol can be reacted with, and things can go on that you might not want. And this will ruin the stability of the DNA when that happens. So the DNA has made the decision, listen, if we can get rid of this, and if we do not need that nucleophile that's out there with that OH group, then we just need to kick it to the curb. We need to get rid of it because it's going to cause us some problems later on down the road. So that's exactly what DNA has done. It has taken this nucleophile and it has eliminated it from the structure. That way it doesn't cause DNA problems later down the road because this OH group wants to make up its mind and decide to react with something. And then before you know it, the DNA begins to unravel and it becomes destable. So the RNA does have it. The RNA does not stay around very long. It's okay with having just that normal ribose that's there. Not a big deal for it. It doesn't really think about it. RNA will go out and it will make proteins and it will tell the body basically what to do. And it's given orders from the DNA molecule. So RNA, not a big deal. It's more easygoing, but DNA has to be particular. It has to stay reserved so that way that the DNA structure can stay around a little bit longer. So it has to stay stable. The OH group helps the stability of it when it leaves, and that's exactly what it needs to do. Okay, so here's RNA. This is just one of those examples here. Here's the RNA structure. There's the sugar backbone, and then here's the phosphorus, and then there's another sugar, and those two sugars are connected. And we see that these OH groups are present. Well, here's an OH group, and this is just one reaction that could go on. So we have a nucleophile or something that could actually come in and rip this hydrogen off, and away it goes. And when that happens, this will make that OH group to be a negative charge. And that negative charge can then turn around and associate with that phosphorus group. And before we know it, this OH group is now interconnecting with the phosphate group, and it's destroying this double bond O. And then this double bond O says, no, wait a minute, I'm going to revert back. And then the phosphorus says, okay, fine, then I'm going to kick this one off. And that sugar can deal with that negative charge. And then we see a cleave in the RNA chain. Imagine that if this happened to your DNA chain. Imagine if your DNA could react with itself and your DNA would start cleaving at different spots along the course of the way and start fragmenting up into smaller and smaller pieces. We would not be here. We would not stay around very long. Our DNA is basically what gives our bodies orders for everything, no matter what it is. And imagine if that boss man was cut down and chopped up into pieces. This company would not run very well because of it, and your body would not run very well either. X-ray crystallography. We talked about X-ray crystallography, and this is the image that they basically generated back in the day. And a lot of people with an untrained eye would not be able to look at this image and make any sense out of it, heads or tails. But what you basically are looking at here is, imagine a tunnel. Imagine you looking down a tunnel, and this tunnel is basically right here in the center. Three-dimensionally, this tunnel is basically you're looking down the train tunnel to the opening at the very, very end, which is in the center. And you kind of see this coil. You kind of see this circular piece that goes around and around and around and around, right? And then as you get closer, you see these darkened images here in the center, and that just kind of keeps that issue going further and further down. So this is a first insight that the DNA molecule made this coil or made this helix as it went down. Not too many people, though, during that time could read X-ray crystallography. And we know Watson and Crick. Crick had some X-ray crystallography knowledge, but he wasn't the one that actually ran these. It was actually a woman. Her name was Rosalind Franklin. And she was a biophysicist, and she was a chemist. So one of the few chemists that was part of this partner, or part of this team, and she was especially trained on X-ray crystallography. The reason that I want to show her is because, number one, she's a woman. Almost every time in all of these lectures, when I watch these videos, or when you watch them and take notes, you're seeing images of men that are up on the screen. And this is kind of the first time that I'm showing a woman that has donated something to science. Well, the issue here is that she was the one who recorded the picture of the DNA. She did that along with another researcher. This researcher took her laboratory notebook, and Watson and Crick showed up basically to the lab one day and said, "We would like to see some of the material that you've taken up so far." So the researcher was basically, "Here you go. Here's Franklin's work that we've done. So just take this and do what you need to do with it." The other researcher, last name of Wilkins, Watson and Crick was awarded the Nobel Prize. Rosalind Franklin was not. Now, was it because she was a woman? That's up to you to decide. But Rosalind Franklin was the one who was running these samples. She was the one that was putting her data into a notebook. She was the one that was interpreting the data and what it meant to people. And the other researcher, when she was not present, basically shared these results with Watson and Crick, and those men ended up with the Nobel Prize, and Rosalind Franklin was not. She passed away at 37 years old. She passed away from ovarian cancer. A lot of people said, well, is it due to the X-ray crystallography? Again, that's up to interpretation. It probably did have something to do with it. Imagine you working around an X-ray machine all day long, every day of your life. There's protection now in place from that, but she did die from ovarian cancer at the age of 37. With that said, before I stop this video today, we also have another chemist that I would like to throw into the picture. And here is our first chemist out of this whole thing. The other men were biologists, and she was a woman. And I told you that most of the pictures up to this point are older white men. Well, here will give me a good chance to introduce this person to you. And this person's name is George Washington Carver. 1864. He was an agricultural chemist. He focused on peanuts and sweet potatoes. Peanuts was a crop that was very important during that time, and peanuts actually replenish nutrients in the soil. So it was this concept that we should start growing peanuts to replenish nutrients in the soil, so that way we can plant other things a little bit later and they'll grow even better. So we could rotate these crops around. So not only did he come up with this peanut plantation, but he also created some peanut recipes that went along with peanuts as you grew them. Peanuts were also used in other products: cosmetics, dyes, paints, plastic, gasoline, nitroglycerin, you name it. Peanut had a use. And he was a pioneer in destroying the stereotype that black race was intellectually inferior to the white race. This was the first time that we get a chemist. This chemist is a black man that we are now giving credit to. 1864. So this was way before Rosalind's time. But a lot of times these people get overlooked and they just get skipped over. And they make quite a bit of contribution to the scientific field. And we've got to give credit where credit is due. And as you can imagine, very early on in this process, George Washington Carver was very important, and he was a leader in the field. But did people really want to give him credit where credit is due? Or did they want to treat them like Rosalind Franklin, where she made a large contribution to science and basically received no credit for it? Think about it. If you've taken a biology class, did you ever mention this woman? Did your instructor ever mention this woman? I guarantee you that they mentioned Watson and Crick. I guaranteed you they told you about the structure of DNA and how important their studies basically were to the scientific community. But at any point in time, did they tell you about Wilkins and Rosalind Franklin? If they did not, then they did not provide these people the justice that they deserve. And they need to be mentioned because it's people like this that made the contributions to science that should not be overlooked. And if this X-ray crystallography did not take place, the 3D structure of DNA would have been harder to interpret. And then who would have done it at that point? Who would have done it? So she plays a very important role in this process. And very often, even in a textbook, they will mention Watson and Crick, and that's really all that they will give credit to. All right? Don't let your textbook lie to you. An author wrote it. which means a point of perspective went into writing it. So don't let them lie to you. Make them tell you the full story. All right, so that's where this video is going to stop. And then the next video, we'll continue on with this 3D structure, and we'll just keep trucking on with DNA studies.